Okay, so last time we talked about the difference between cells. This time we talked about the organelles and the different types of organelles they have. The first one is the cytoskeleton, and we kind of talked about that in the last video. It's what gives it. It's like our skeleton, except it's for the cells. So it supports and gives shape to the cell. It also helps organelles move around. It's kind of like a a rail system, kind of where they can kind of cruise around. And it helps when we talk about cell division. The cells dividing into other cells, and for cells that have flagella and can swim and move stuff, it helps for them too. So that's cyto one. When cells, what they need to do is they need to make proteins to be able to do stuff. So to do that, you need to start out with the instructions, and the nucleus stores those instructions in the form of DNA, and that's one of the biggest things the nucleus does. Now it sends out its information through, you can see these pores here. It'll make copies of the DNA, but it'll make it in terms of RNA, and it'll send it out to. Lots of stuff happens in the next part, the endoplasmic reticulum. You can see it looks really wavy, and I'll show you other pictures of it that are better. There's some rough ones that have all these ribosomes on them, and that makes it rough endoplasmic. Here's a better picture of it. So the nucleus would usually be right in here somewhere. This would be the nucleus. This would be the rough endoplasmic reticulum, and this is the smooth endoplasmic reticulum. You can see it's rough because it looks bumpy, and it's smooth because it's not bumpy. The bumps are ribosomes, so it's very important to know what causes the rough. Now the nucleus, like I said, is the control center. Right? It has the DNA, and most of it is in the nucleus. We'll talk about later. There's some DNA that mitochondria and chloroplasts have. They have their own DNA, which is kind of interesting, and it's kind of a cool thing. But we'll talk about that a little bit later. The nuclear envelope is the like gatekeeper around, or it's the fence around the nucleus. It's what keeps the DNA out of the cytoplasm and protects it from all the stuff that's in the cytoplasm. But things do need to come in and out, and they do through nuclear pores. Nuclear pores are those holes that allow things to go back and forth. Cytoplasm, we know what that is. Nucleoplasm, same thing as cytoplasm, except it's inside the nucleus. That should be pretty obvious. And the nuclear lamina is just a way of organizing it, the stuff inside the nucleus. I wouldn't worry about that too much. I'm pretty sure that's not on the test. Now, when a cell divides, when we need to make new cells, it should be kind of common sense that, well, if we're going to have another cell, we need to have just as much DNA for both cells. So we need to copy the DNA or reproduce the DNA to be passed on to the cells. And that's called DNA replication. And that takes place inside the nucleus also. As we already know, DNA are sequences of nucleotides, right? DNA is the polymer. Nucleotides, individual nucleotides, are the monomers. Excuse me. The nucleus transcribes, is what it's called, when we change from DNA into RNA. And this is the stuff that goes out into the cell. So we have these original copies of DNA that we keep safe in the nucleus. We make little copies of the RNA that go out to make the proteins. Right. If these get ruined, that's no big deal. We can just make more copies. And that's how that works. These go through, they're made inside the nucleus, they go through the nuclear pores, go out, and they'll find ribosomes, which we'll talk about in a little bit, and makes. When we have DNA, and it's all coiled up inside our nucleus, it's called chromatin. It's just different names for it. If we look at all the chromatin and all the individual unique structures, they're chromosomes. So the chromosomes, usually when we draw chromosomes, you'll see them, they kind of look kind of like this. And then if it has its pair, it looks like that. Sometimes you'll see them like that. Each human cell has 46 chromosomes, 23 pairs. Right, we got 23 from each parent. Now, inside the nuclei, usually you'll see something called a nucleolus. And that's the really dark region in there. And that's where RNA is made that's for ribosomes. This is RNA that's used in ribosomes. Which ribosomes are the little things that we showed on the endoplasmic reticulum to make it rough. They are what makes proteins. So Ribosomal RNA is made in the nucleolus. The proteins needed to make these ribosomes are synthesized in the cytoplasm. So it kind of goes back and forth, right? The proteins that go back in there to make ribosomes are made outside, and it's kind of confusing, but you just got to remember that it goes kind of back and forth through the nuclear pores. Enough about ribosomal RNA. We'll do a lot of that later. Back to the smooth and... Why is this picture back here? I don't know why that picture's there, but oh well. Two types of endoplasmic reticulum, or ER. We like to be lazy, rough, and smooth. The ribosomes make proteins by putting the amino acids together, and they join them with, everybody remembers this, of course, peptide bonds. Peptide bonds. They make polypeptides. Remember, that's another name for that stuff, polypeptide. I'm going to run out of room, but you get the idea. There's vesicles also, which are little, they kind of sound like vehicles when we say it, and that's another good way of thinking about it. These vesicles move through the cell and they carry stuff. So all these vesicles right here, similar to V stuff. Ribosomes, they're the organelles that can be floating around free in the cytoplasm, or remember they're attached to the ER to make the rough ER. They're made out of RNA and proteins, which is kind of a unique little thing. They contain the enzyme that forms the peptide bonds. We said that. Each ribosome is it's made out of a large subunit and a small subunit. Don't worry about that. It is a big deal, but not for this class. The ER, the endoplasmic reticulum, it's a bunch of flat sac-like structures that form compartments. On the inside, the ER lumen is called the inside, and that's between the two membranes of the nuclear envelope. The two surfaces of the ER have different enzymes in them, and they have different jobs. There's two different types. There's the rough ER and the smooth ER. The smooth ER looks more tubular. You saw that picture with a smoother outside surface, which is why it's called smooth. The enzymes in there are really involved in making, catalyze means to make, lipids and carbohydrates, especially phospholipids, which are in the cell membrane, right? Cell membrane. Same as cholesterol, that's also in the cell membrane. The smooth ER also breaks down glycogen stuff and detoxifies dangerous chemicals. So that's something that helps get rid of poisons when we eat poisons, is our smooth ER. The rough ER is rough because of the ribosomes. We said that. When proteins are made on these ribosomes, they're usually sent out of the cell. They'll either go to different organelles or they'll go exported from the cell. They'll get sent out of the cell. The Golgi. This is one of the coolest ones. Sometimes it's Golgi complex, sometimes Golgi body, sometimes Golgi apparatus. All those are pretty much the same. The big thing you need to remember is the Golgi part. Right? And it's a bunch of flat sacs also. This has two different faces too. It has the place where they go in, called the cis face. A lot of times cis is thought of as the same side, if it's a cis, and trans means opposite. So if it's the same side, that's where it goes into the Golgi, and that's the cis. Trans is the opposite side, where it goes out. Medial just means in the middle. They make a lot of glycoproteins. So what happens in there, besides being like the shipping area for proteins, it'll make it a glycoprotein, and that's just adding a sugar or a carbohydrate. That's adding a sugar to protein. The transport vehicles, or vesicles, sorry, I even messed up thinking. I always try to remember vesicles as vehicles. We'll take proteins from the cis phase and move them along the Golgi apparatus. The Golgi apparatus is like the processing section, kind of like FedEx or UPS. It'll process, it'll sort, it'll actually change them. So hopefully UPS isn't changing your package, but it'll definitely process and sort it, and then it'll send it on its way with the transport vesicles, again, leaving the trans phase. So it goes in the cis phase. So if we have, here's the face, this is cis side, this is trans, right? Goes in here and out there. Some other important organelles. Obviously, mitochondria. We need energy in our cells. So mitochondria is the thing that the sugar that we ate, the chemical energy, like glucose and turns it into ATP. Remember we learned about ATP last time. It's a nucleic acid that has lots of energy. The energy is in the TP part, the 3 phosphates. These bonds right here between the phosphates have lots of energy right there. So those are where all the energies are. There's also one that we kind of think of as energy as chloroplasts, and those are the ones that take the energy from the sun, right? And they convert it to this chemical energy, or the glucose. Something interesting about them is mitochondria and chloroplasts both have their own DNA, which is really, really weird. It really supports the idea that maybe we got these organelles through something called endocytosis, where a different organism, like a microorganism, was fused together with another one, and it kind of stayed in there. And that's why those different sets of DNA are inside the mitochondria and the chloroplast. This is kind of cool. Look, it looks like I wrote poop. I didn't write poop, by the way. That's phosphate with... Never mind. Okay, anyways. So the mitochondria is where we break down the sugar and energy, and that's called aerobic respiration. Aerobic means we need air, and it makes ATP. It takes place in the matrix, which is the space inside of the mitochondria. If you remember the mitochondria, it looked kind of like this, and then it had all this stuff, right? There are actually 2 membranes in there, right? This is the inside, and this is the space in between the membranes. This is the inner membrane space. Inner membrane. And this is the matrix. Not where Neo is. That's a movie. Right? So it's not there. The folds are called cristae. Don't worry about that. I'm not going to ask you anything about that one. And the inner membrane is where the ATP is made. That's going to be really important when we get to that chapter, which is the next chapter. Some other organelles, vacuoles. Those are usually in plant cells. You'll see them. You'll see a big vacuole that helps kind of, and that's what vacuoles do. There's a large one usually in plants that are really, really important in keeping the plant solid and making it not be all floppy. If you watch or if you look at your plant and you haven't watered it for a long time, you'll see that it kind of will get all loose and fall over. Well, that's because your vacuoles are running out of water, so they store water. Give it a bunch of water, and within a few minutes, the plant's all standing tall and stuff. Lysosomes. These things are cool. These have lots of enzymes, which, remember, we talked about enzymes are proteins that speed up reactions, chemical reactions. Well, one of the things they do is digest different materials. So these lysosomes are little sacks of things that go around and chew up stuff. Right? They're important in maintaining, or they maintain, a pH of about 5. So they're very acidic usually. Right? And they go around chewing up invaders. They chew up old organelles that don't work anymore. They're kind of like our garbage truck. Also centrioles. These centrioles are only in plant cells. Did they put that on there? Animal cells only. They're not in plant cells. Animal cells only. And these are really, really important for helping divide the DNA. They actually grab onto the chromosomes and pull them apart. They also help form the cilia and the flagella, which you guys know are the little hair-like structures. Or cilia are the little tiny hairs, and flagella are the long tails. We talked about that in the very... Plant cells also have something different in that they have cell walls. Besides a membrane, they have this really strong support it. They also have chloroplasts, which we talked about earlier. Those are the ones that take the energy from the sun and convert it to sugars. That through something called photosynthesis. So that's taking the light energy and converting it to carbohydrates, especially glucose. Now, chloroplasts have chlorophyll, and that's what gives it the green color. So the chlorophyll is the molecule or the pigment that gives it the green color. They also have those carotenoids. Remember we learned about those last chapter? Those are a type of lipid that we learned about. So what they have is they have these cool little sacs in them, like this. Right, like this. Each of these sacs is called a thylakoid. Where is that written? Here it is, thylakoid. Now, if we take a whole stack of these little sacs, it's called a grana. Right? And all this stuff is inside of the chloroplast. And this is where all the cool stuff happens. When we get to that section, there's lots of cool stuff. So inside the thylakoid membrane, the chlorophyll molecules absorb the energy from the sun, excite electrons. Remember, electrons get move along this thing to produce ATP. Remember we talked about NAD plus and NADH. That's where these things are getting all crazy and doing all the work. And they're moving the electrons around. So this electron will come here attached to that. It'll become NADH. It'll go back over here for the sunlight to excite it again, and it starts all over again. It's pretty cool. And we'll learn that in detail later. All right, so that's pretty much it for the organelles. Now you have all your notes. That didn't last very long. That was pretty quick. All right, bye.